When you give a broad spectrum antibiotic, it's killing everything. It's carpet bombing, and it's killing the beneficial microbes as well. Which, okay, if you want to get rid of the pathogen, like if you have a urinary tract infection, you have an E. coli, you want to get rid of that pathogen. But then why not at the same time give a probiotic, or afterwards for another month give a probiotic that tries to help the person's beneficial microbes that you've just wiped out, tries to bring them. I should also say, if you also take fermented food, that to me is a benefit, because fermented foods like kimchi, kombucha, and these things, they have a lot of beneficial microbes in them. They're not probiotics, but they have beneficial microbes in them. So I encourage people to take fermented foods to help replenish those organisms. So it's not just a case of taking a probiotic pill. You could be eating fermented foods at the same time. All these kids' antibiotics, so there's multi-drug resistant organisms. How do we get beneficial microbes into that environment? The only way is through a probiotic. Now, you could say the same in a hospital, a ward, or a place where patients are. Why on earth are we not doing that? We're very happy to give the toxic drugs, and yet we won't give beneficial microbes. So I think we have to get people to do that.
Hey everyone, Rupesh Patel here, host of the Two Nobodies podcast, and you just heard from Dr. Gregor Reid, who's considered one of the pioneers and foremost thinkers on the topic of probiotics. In this episode, I get curious about the use of antibiotics, the detriment that it's had on the gut microbiome, and can probiotics actually play a beneficial role? I've had many conversations with a number of researchers, and it's been unclear to me whether probiotics have a beneficial effect. Dr. Gregor Reid and I go through all of this. We also go through some of the top search questions that folks have had on Google and on YouTube about probiotics, so look forward to that. We go through some of the myths and fallacies and some of the things that you need to consider as a consumer when you're thinking about a probiotic. What about the role of doctors in the regulatory system? What kind of things can governments do to ensure that probiotics have some sort of legitimacy? It's a fascinating chat about all things probiotics with Dr. Gregor Reid. I hope you enjoy this episode. Like, subscribe, and I look forward to all of your comments. Cheers.
Welcome to the Two Nobodies podcast with my dad.
Gregor, so lovely to have you on the show. I've been thinking about this topic for some time. For folks who don't know, I've actually recorded, I feel like I could turn this podcast into a bit of a microbiome or gut health podcast because it's such a fascinating topic. I've talked to Dr. Alan Verco a few times, talked to, I think, your former PhD student, Brendan Daisley, about honeybees and the microbiome and probiotics there, and also David Good talking about the Yanomami peoples, and his research there was just fascinating. One of the areas, though, when I talked to both Emma and Brendan, which really triggered me to follow up with you, was the conversation with probiotics, and learning from Emma that there isn't any sort of colonization that happens with probiotics. And so I left feeling that conversation. I'm like, okay, maybe probiotics, there isn't really anything there. And then I talked to Brendan, and Brendan's like, well, yeah, that might be true, but just because it doesn't colonize doesn't mean that there isn't any benefits to probiotics. And that was like, okay. And he was like, you got to talk to Gregor. He is the person on probiotics. And here we are. So welcome to the Two Nobodies podcast, Gregor. Looking forward to our conversation today.
Thank you. It's nice to meet you.
Yeah, it's really great to meet you. And I know you have this book, which I did buy recently. I've started reading, Gregor, so it's been an interesting read so far. And definitely want to make sure that folks read up on that book there. But let's start off with that first piece about how with probiotics, I think the common thought is that it will colonize in the gut, that you get all these beneficial microbes, and that it's going to maybe stay there. And that's sort of how it works, but that's not quite how it works, hey?
No. I mean, the only possible exception might be if you gave it to babies, and because they don't have their own microbiome formed. There might be an occasion where a strain might colonize, but there's not really been much evidence of that either. So the human has its own microbiome that has developed over time. And it's a bit like you go to a stadium to see a game and all the seats are taken. They might let you in and with a foghorn and orchestrate some chanting or do something, but you're not going to get a seat. And so it's still worthwhile having that person come in and leading the songs, especially if you're a Scottish football fan. There's lots of songs, but we don't know fully why they don't stay. I mean, there's the concept that there's already organisms there and they don't need new ones to stick around, but they don't. So the point of taking them regularly is because they don't stay. And still to get the benefits, because they multiply and they produce compounds, or they do things in the gut or in other places, the mouth, the vagina, the skin, etc.
Right.
And so it's a bit of a myth that they colonize. I mean, there are papers showing some strains have stuck around or persisted for quite a while. But yeah, in general, they don't colonize. Persistence is a better term because they could persist for days or weeks or months, but in general, they don't colonize. Yeah.
And the microbiome is from what I've understood is it's a bit of a community, right? Like they, you know, and like you said, you know, your analogy to a football game, a soccer match, where all the seats are full, they may or may not like these kind of new microbes that are coming in, and there may not be any sort of symbiosis there to kind of allow these microbes. Is that fairly accurate?
Yeah, and it is a puzzle because so I always think of it's a consortium, and I sometimes say to students, if you go into a class and you teach the same class, say 5 days a week, you often find that certain people sit beside certain people, and you look at them and say, well, why are you sitting next to that person?
Right.
And most often it's because they're friends, but sometimes it's they're sharing lunches or whatever. And so in these microbiomes, the organisms have developed into a structure and a cooperation or coexistence. Sometimes they actually need each other, as Emma Allen Burko's work has shown. And the mouth, the oral microbiome, is a guy called Colin Brander. He has shown quite nicely that it's the streptococci come in first, and then the secondary colonizers and third colonizers. So it's kind of organized itself. Now, you might wonder if it has a lactobacillus, say rhamnosus, that's part of that consortium, why wouldn't it accept a probiotic lactobacillus rhamnosus?
Yeah.
And I don't think we really fully understand why they're rejected or not allowed to be part of it. Now, you might also think then, what about if you have serious disease, say Clostridium difficile, and everything's flushed out, then the fecal microbiota comes in and it does stick around. And I think that's because everything else has kind of been taken away. And the consortium that came in is in this form where they're cooperating with each other. And so it is a puzzle to me. I don't really have the answer for you as to say why a probiotic strain would not stick around, even if its uncle or aunt, was there already. And you look at them and say, well, they're both the same species, the same genus, and why aren't they staying there? And maybe in part it's because of how we deliver them, because we deliver them usually as dried organisms. Maybe it takes a while for them to get up and growing, and then maybe there's things about them that they don't, they're not able to become part of that microbiome. I think that it's going to take some research for us to understand it.
And especially like if there's a microbe missing and we try to introduce that strain, you'd think that there should be some acceptance there, but even in that case,
and I think especially when someone's been on lots of antibiotics, you've really disrupted that microbiome. And so why wouldn't it accept another strain? And, maybe it does in some occasions, but in general, we're not finding these probiotics sticking around.
Is there a way, can the research help actually determine why these probiotics are not sticking around?
Yeah, I mean, I think... there's a guy called Swidzinski. I think he's German, and he did this really neat study in the vagina where he basically labeled the organisms, with
Yeah.
fluorescent labeling, and therefore you could look at where are they positioned in that area, right? If we could do the same in the gut and say, okay, why is this particular species or genus pretty much always beside this other one? We know they're there, but we don't know
Yeah.
where they're located. We don't know who they're binding to and who they're interacting with. That's going to take, I mean, imagine if you had, 1,000 different labels, that's almost impossible. But even if you started with 20 or 30 labels on genuses, or genera, we should say, then you could kind of start to see that pattern, because the genome sequencing isn't telling us that. It's just telling us what's there. So I think we need to understand that. And then we say, okay, in a person who maybe has diseases or inflammation or something not quite right, maybe we know that there's more of a certain phylum, a family of bacteria, but again, we don't know what these interconnections are and how you break up those interconnections. And in fact, maybe in the future, it's not an antibiotic you need to kill the organism. You need something to break down their connection with the other organisms that are there. That potentially could be a new way of treating these conditions, because, I mean, if you come in and there's 50 organisms and they're all together with other strains, and you kill 10 of them, well, as soon as you stop the antibiotic, back come the other 40, they're going to multiply and you're back to square one. It's very difficult to specifically just get rid of a single pathogen. But maybe if you change the environment in such a way that they're no longer connected to the other organisms, maybe it just makes a place that they don't want to stay in. I mean, very, very hypothetical, but I think that's the kind of approaches we need to start to take.
And do you know if folks are trying to understand this question?
I think there's people trying to look at the linkages of organisms, but I haven't seen anything in terms of labeling. I mean, I would essentially just take a sample from the gut, multiple labels on it, and just look. I mean, it's a needle in a haystack, but we really don't understand which ones are feeding off the other one, or why they're there for a particular reason. And if you think about it, we all have a different microbiome, and yet most of us there's a similarity of function. So even with a very different microbiome, the functions seem to be the same, which again means that there's different types of organisms that are interconnecting so that they can digest the food, they can modulate immunity, they can do things for us, but it's a different subset that you have compared to me, perhaps. And so we don't understand that. What people are looking at is, okay, in your subset, is there some organisms that really are quite nasty and down the road are going to get you in trouble, in which case we need to get rid of them? Or are there some that are in low abundance that we need to give a prebiotic to in order to stimulate it and bring it back up? So I think people are looking at it from that way, but I don't know of research that's saying, why is your normal and my normal quite different, and how are they functioning in such a way that we don't really feel any symptoms or signs? There's nothing saying that we're in bad shape. I haven't seen that kind of work.
I know when I spoke with Emma, I was asking her about, how far has the research come along? Like, why haven't we been studying this for a while? And she's like, it's really just been in the last sort of 15, 20 years that we've been making some serious breakthroughs. Some of it has to do with the technology that's out there. But when reading, the first few chapters of your book, like, you talk a little bit about the research, and, you sort of starting in this maybe in like the 70s or 80s, and no one really wanted to even entertain the idea of these kind of beneficial microbes. So interesting to hear your perspective, minus the technology piece that kind of Emma's talked about. But even before that, like, why weren't people really thinking about beneficial microbes and probiotics? So what were some of the barriers that you felt like you had to overcome there?
Talk about barriers, that'll be a long discussion. So if I give the example of Dr. Andrew Bruce, who in 1973 published a paper. So he looked at women had recurrent urinary tract infection. They found E. coli in the bladder and E. coli in the perineum and the vagina. In the women who were healthy, who never had a urinary tract infection, he found lactobacilli. So back, if you look back in the 70s, pretty much everyone, and some of the people actually went on the Nobel Prize Committee because their work was deemed, interesting. Everyone looked at E. coli, and they tried to come up with a vaccine, and they tried to develop ways to counter the E. coli.
Mm-hmm.
No one cared about the lactobacilli.
Mm.
And so I joined them in 82. So that's almost 10 years later. So you can imagine no one had done it. And then by the time we started looking at it, it was like, you're joking. Why would you be looking at these organisms? They're not doing anything. And we have antibiotics for E. coli. We don't need work on lactobacilli.
Mm-hmm.
And if you look also at the grants panels, certainly in Canada, they were all filled with people that studied pathogens. It's kind of ironic because it's the Canadian Institute for Health Research. It should really be the Canadian Institute for Disease Research. But anyway, they were filled with people. And one of the chairs of the committee basically told me at one point, don't send your grant in here. We have no one to look at probiotics. We're not interested in it. So the system itself was not saying, let's look at essentially microecology. And it's no longer just the gut. I mean, there's people studying, as you said, for Brendan, for insects, and people are looking at coral and fish. And there's this, the Human Microbiome Project opened this up because they realized that there were other organisms, not just pathogens. Mm-hmm. We had, of course, shown it in the vagina for many years, but nobody paid attention. So I think that delayed, by 20 to 30 years, this type of work. Wow. I mean, there was always a microecology set of scientists. They had their own meetings, and, it was, I would say, they did a lot of really interesting work, and they became pioneers really in this field, but nobody was excited about it. And if you go to a medical conference, no one was looking at this. If you go to even microbiology conferences, there wasn't enough on the beneficial microbes, and that's completely changed now. There's societies that have beneficial microbes as the core. So I think the other thing was in industry. There was no interest in industry. Industry just wanted antibiotics. Yeah, and in fact, it's ironic because we funded some of our work through essentially money that was hiring people, but we would get the people to do the work that the company wanted on an antibiotic, but we'd also get the people to work on other things that were lactobacilli. We studied all kinds of antibiotics, and that's where the money was. Now, of course, it's not there either, but back then it was. And so industry was not ready at all. The regulatory agencies weren't ready either. So the whole system was set up for pathogens and disease.
Well, and like you said, if none of those grant panels had the right people to be able to even evaluate these grants, then you had no chance, right?
No, and it's kind of ironic because every year there's boards, advisory boards for the different within CIHR, and they often I was asked to put my name forward. Never once was I considered to be on these boards, and so the boards are influencing the committees and who's on the committee. And so therefore, as I said, the whole system, it didn't want input from someone like me. And therefore, it was very, very hard to get grants. And that slowed the whole process down.
How did those boards get established? Like who's responsible?
Well, that's a good question because, again, it's the hierarchy of CIHR in that case.
Yeah.
And, I've seen it, a person would be a chair of a board or chair of a panel, and next minute their favorite postdoc or favorite PhD student is now taking their place. And so you get this kind of
the family business.
I felt it was a bit of a boys' club, to be honest. And I know a lot of women who didn't get funded when they should have on women's infections like urinary tract infections.
Right.
So I think the system really needed drastic change, and it never did. They'll argue, of course, now that, we accept everything, blah, blah, blah. But the proof is in the pudding. I mean, how how many grants have really been funded on beneficial microbes. In the last 5 years, probably more, but before that, 30 years before that, hardly any.
Yeah.
And people have had their career studying salmonella or E. coli surface membranes. I mean, good for them, but how has that helped humanity? I don't honestly know. But there's been millions and millions of dollars spent on it.
When did your research program start to take off then? Like, and I guess what sort of enabled that? Because you have over like 600 or so peer review publications, so there's a good body of research there. Like, when did it really start to take off, and what was the catalyst, I guess?
Probably around 2000, 2001.
Okay, so not that long ago.
No. And we had done some clinical work, and we got this massive grant from a company that really allowed us to do some clinical trials, allowed us to do research and publications. There was more papers came out. I think then there was also, I think more students became interested. It's funny because it's like in urology, if you're studying prostate cancer, you'll get lots of students because the urologist will then become a urologist. So a resident would become a urologist, and then they would be able to operate on the prostate.
Yeah.
No one was interested in urinary tract infection. You can't. It's not really. It's not a moneymaker. You can't really operate. So as a surgeon, why would you be interested? And so you have this kind of disconnect in terms of urology, at least. But in terms of microbiology, it's a bit the same. It's kind of sexy to study COVID. And you look at the millions and millions that went into COVID in Canada. We even applied for a grant, and they said we didn't have an epidemiologist, so therefore we couldn't do the work. It was like crazy, but that's another topic. But if it's not sort of sexy stuff, you don't get the funding. And lactobacilli has never really been very sexy. So
fair. Yeah. What throughout this whole time, what's really been that driver for you, kept you curious? Like, is it, I know you talked about sort of, you said you've talked about people research E. coli and make careers out of that, but you just sort of talked about how does it serve humanity? I read in the sort of first couple of chapters of your book, there seems to be this real interest on your hand of wanting to serve humanity and really have that sort of broader positive impact. Is that sort of the driver, or was there this fascination with these beneficial microbes, or what's really the driver there, Gregor?
Yeah, I've always been interested in application. I think there's clearly a place for basic fundamental research because you never know where the breakthroughs will come from. But I've always felt that you have to apply the work, especially if you're saying that you're working in the human health field. You have to somehow apply your work to the betterment of that. Now, maybe it won't get to clinical trials, but at least you can do studies in humans. I think the funding agencies have not done enough to support work on humans. You can easily get a grant if you study mice. I've never been a fan of mice. They're so different from humans. And so I think much more money should have gone to human research. So I've always been interested in that. That's my own preference. And I guess through that, you can also look at mechanisms. I mean, the GR1 strain is probably the most researched strain of probiotics that's out there in terms of looking at the organism itself. Lactobacillus GG has the most publications. Many of them are clinical studies. But in terms of understanding the organisms, I think we were probably, GR1 strain was probably the most, because we were interested in finding out what it did. And so the thing that drove me was I felt we could somehow make a difference to women's health. And if we didn't, then we'd die trying. And then, of course, the work in Africa and other places was because I think everyone should have access to these types of benefits, not just people who have money. So that's a whole other different topic. But yeah, I mean, initially I wanted to do medicine, and I didn't get. I had the marks, but I didn't get into the program. And it turns out that in Glasgow University back in 78, the headmaster of my school found out that people with lower marks than me got into medicine because their father was a professor or a medic. So those were the days. And so what that does to you, it gets you, you either give up or you become a fighter. And I became a fighter.
Right.
And so I was determined to do something that was impactful. And that's what drove me.
Sounds like you experienced a bit of nepotism early on then, hey? And then like you talked about like some of your research applications and not sort of, allowed to be at the table kind of thing. Oh, that's fascinating. That's a good segue.
Like part of the reason I left Scotland, there was also a class system too at that time. I mean, people would ask you, what school did you go to? And I went to a school in a pretty rough part of outside Glasgow. Okay. So and when I came first to North America, it was in 1974. It felt like they just accept you for what you are here. Right. And so that was one of the things that drove me out of Scotland, really, and into Canada.
Because you came to Calgary, is that right, first? Or
yeah, well, I went to New Zealand for my PhD and then went to Calgary. But I was to set up a project in Halifax and another one in Toronto. And when I got to Toronto, I met Dr. Bruce. Yeah. And we just hit it off. And so I called my boss in Calgary, Bill Costerton, and said, do you mind if I'm based here doing the work rather than based in Calgary? He said, sure. So that was a lucky break for me.
Yeah. That's a good segue to this book, Probiotics Story About Hope. I
made it a story because I didn't want it to be facts and figures and tables and graphs, and I wanted it to be a story. So
and you weave in some golf analogies in there.
I don't know why I did that, but it just a lot of golf really is facing up to challenges and trying to overcome it. So
yeah. And so in this book, my understanding is you're trying to sort of clarify folks really what probiotics are. You're talking about just, probiotics for women and supporting women's health. And then I think some of it is also to shine a light on all the misconceptions or all the different things that, companies are doing when it comes to selling probiotics and all the issues there. Is that sort of the main objectives that you're trying to get across?
Yeah, it was. I was getting fed up with companies getting away with saying, and I gave lots of examples like probiotic aftershave and probiotic mattresses, and I mean, it's just totally ridiculous. And it's no wonder some of the medical community are cynical about the field because people are saying stupid things about it. So that was a big driver. Also, to tell the story and bring attention to women's health and some of the things that we had done. And then also to tell kids that even if you have a brilliant idea and everybody says you're an idiot, don't necessarily believe them. If you truly believe in what you're doing, give it your best shot and don't give up. So it was kind of almost a business story, but a mentorship story as well.
I love the way it's reading. Let's start with probiotics itself in terms of the general elements that make up a probiotic. Like, how do you describe that to sort of the layperson? I know it's a bit tricky, but how do you typically describe that to a layperson?
Well, they have to be live organisms. Okay. So if it's a dead probiotic or if they're dead at the end of shelf life, then it's not a probiotic. They have to be administered. So we used the word administered when the definition came out in 2001 because everybody thinks of probiotics only that you swallow them. That's not the case. You can have probiotics that go in the mouth. You can have them in the vagina and on the skin, etc. Yeah. And in adequate amounts, that's important to me because it seems like when you look at adverts, they say we've got billions and billions, and so we have the most of any product. That's not relevant. It's how many do you need? And have you done a study to show that you need this amount versus this amount? And very, very few people have done comparative dosage studies. We did some. We were one of the rare ones. Inadequate amounts conferring a health benefit. So how do you prove you have a health benefit? Well, lots of these websites have got testimonials. testimonial doesn't prove anything. It's it's nice that people say things nice about the product, but what's the proof? Is it better than placebo? Is it better than standard therapy? Are there trials that have proven that? Because if the thing has not been proven, then it's really not a probiotic, and that's very hard for people to get their head around. And one of the nuances of this is if you have Lactobacillus GG, which is a probiotic, say for helping prevent diarrhea, or and you then put it in a product for vaginal health, it's actually not a probiotic for vaginal health. So you shouldn't be calling it probiotic if you're putting it in a product for vaginal health, because you have to prove that it's a probiotic for that health benefit that you're claiming it has.
Yes.
And so therefore what happens in industry is they go to a manufacturer and they say, give me a strain of this and this and this. And the company sends them, and then they put it into a capsule or a tablet form and they sell it. And they're relying on the company someone having done the research on that strain to prove that it has benefits. In most cases, these strains have not been proven to have benefits. They shouldn't be called probiotic. In other cases, if I keep going back to Lactobacillus GG, but if GG at 10 to the power 9 has been shown to have a benefit, and you put it at 10 to the power 3, it's not going to have that same benefit unless you've proved otherwise. So another example, which I give in the book, is inulin. So inulin is a prebiotic if you take 5 grams of it. But if you put 1 nanogram of inulin in your product, it's not a prebiotic. Don't call it a prebiotic. So for lay people, it's very hard to get their heads around that, because they'll pick up a product and they'll say, this has a probiotic in it, and there's no real evidence that it confers any health benefit. And this has inulin in it, therefore it has a prebiotic in it. Well, no, you don't have enough inulin in it to have a prebiotic effect. So in fact, you shouldn't be calling it probiotic, prebiotic, or symbiotic. That's hard for people to understand.
So probiotic has to be live, has to be administered, has to have an adequate amounts, and it has to have a proven health benefit. On the adequate amount, so I understand, like you said, the research has to show exactly how much is really needed to get that proven health benefit. I would imagine, though, adequate obviously differs from individual to individual, though, too, right?
Yeah. Well, part of it is a practical thing. Like if you're making a capsule that people can swallow, you're already limited by that capsule size. So in other words, if you wanted to deliver 10 to the power 11, you may need to have a sachet and more organisms, or you need to take more capsules, a lot more capsules. So there's a practical side, because most people will take 1 or 2 capsules a day, or at a time, but they're not going to take 26.
Sure.
So and also the price goes through the roof if you have higher numbers. So it actually makes sense to just provide the numbers that are required. And in some people, yes, you're right, it might not be enough. In other people, it might be just fine. That's that gets down to individualizing a probiotic, which it would be great to have that, but practically it's pretty much impossible.
Yeah.
So you do enough of studies of enough different types of people to say, okay, in general, if you look at 100 people or 50 people, this dose will give you an effect that we think. And if it doesn't, either you can double the dose, or maybe this particular probiotic is just not having an impact in you.
So that's just someone playing around with it until they kind of get the desired result that they're looking for, perhaps.
Yeah, I mean, it's like everything. Sometimes you can take 1 Advil, and sometimes you need 2. Yeah. And it's easier with drugs because the levels are easy to control, and they've done enough testing to say that you can take 2 Advil, but don't take 3, because then you start to get side effects that we don't want you to get. In probiotics, there's not really an upper limit. Your body would soon tell you if you overdid it, probably. And I remember one woman who took this particular yogurt. She says she took 3 yogurts thinking that was better for her, and she got the runs. and her body was saying, look, you don't need to take 3. It's not gonna, it's not helpful in your system.
Yeah. I saw you share some links in the book about places that folks can go to find some of these probiotics that have been, that have proven health benefits. I went to Probiotic Charts. Is really, really interesting. That seems like a good place to kind of start for people. Hey,
Yep.
but even then, are there any cautions that you would have if people went to these and be like, oh, well, this one looks like it could help with IBS, or this one can look like
Yeah.
it can help with weight loss or whatever. From a practical standpoint, are there any cautions with just for them to go pick up that particular commercialized probiotic and take it, or?
So the nice thing about the chart is that if a company has not done clinical studies, it's not in that chart.
Mm-hmm.
So if you go to a health food store and say, oh, this product X is absolutely unbelievable, then
Mm-hmm.
if it's not in the chart, then you have to say, well, what human studies have been done on this product or the strains in this product?
Yeah.
And if the answer is none, then I wouldn't even consider it. So then you go to the chart, and for example, if you have IBS, there's different types of IBS. There's diarrhea, constipation, and there might be 3 options. That might come down to maybe one is a food option and you prefer to take it as a food. Maybe it's a capsule option and there's 2 different types of products. And maybe you just have to try one and see if it helps you. And if it doesn't after a month or 2 months, then maybe you have to try the other one. So it gives people an option. Also in the chart, it looks at level of evidence. So some have a very high level of evidence. This is a very good, it's not only a good product, but it's got a good chance of working. Others,
Yep.
the clinical trial data is maybe not as strong as it should be, in which case you should maybe not consider that as your first option. But the charts certainly help. Of course, in terms of bad outcomes, I don't really think that I guess the only thing I would worry about would be if someone thought they could rely on a probiotic or a fermented food even, which isn't on that list, rather than going to the doctor. And that particularly worries me with mental health, because there's a lot of hype around the gut-brain axis and products that help your mental health. And if somebody really needs to have an SDRI or a drug to help them with mental health, they're not going to get that effect with the probiotic. So I think that's my only worry, that people think these are magic bullets. I think in many cases they're supplementary, they augment things. If you're a woman who keeps getting urinary tract infections, I think probiotics could become a first option, because there's 1 or 2 studies showing that if you take an antibiotic, which is the standard treatment, they give low dose antibiotics, that isn't particularly helpful in the long term. So in that case, maybe a probiotic could in fact prevent you getting an antibiotic.
Yeah.
You would hope that the urologist or the family doctor that you went to would study that literature and give you the right advice. But I guess my point, again, going back to say Crohn's disease, probiotics will not prevent or not put you into remission. And so clearly you go to a doctor and they would give you a biologic, and hopefully that would help you. So I think that'd be my only caution. It's there's still we should be looking at traditional medicine as well.
On the definition of the probiotic, to me, I mean, that makes perfect sense. Is there, and I'm just not aware of this, Gregor, but in terms of the regulatory landscape, is this definition not regulated then, I guess, so that commercial producers would have to make sure that it aligns with that definition? Like, what are we missing there from a regulatory perspective?
Well, the FDA don't use the definition. They see it as a biologic or biotherapy. It's I personally find that regulators very frustrating. If you look at the history of regulation, they were brought in to stop literally snake oil being sold over 100 years ago. And then they somewhere along the line, maybe it was influenced by drug companies, I don't know, they decided that only a drug could prevent, treat, cure, mitigate disease.
Mm-hmm.
Well, that's not true. You can prevent diabetes with exercise and the right diet, etc. So food, in fact, does help prevent disease, and the microbes can be part of that. So I think the regulatory system has over all these years needs to be changed. That the Deshea Act in the States at least allowed for dietary supplements to come in, but it would take an act of Congress, or in Canada, an act of government to change it. And in my opinion, we should have a separate, complete entity for microbes, because they're not drugs. The people in regulatory, for the most part, have been used to analyzing drugs, drug safety, drug composition, etc. That's not what microbes are like. So there are structure function claims, but they're pretty general. And so you'll see products that will say good for gut health, but they all say that. I mean, and then Europe, they got to the stage where they almost banned the word probiotic, because they said, oh, you have to prove a health benefit and your trials aren't strong enough. And so they went completely way too far. They had a panel that was the wrong panel for this, and they had literally no probiotic expert on the panel. So I think regulatory, probably Canada has done the best, at least trying to have some claims and trying to adjudicate this better than any other country. But there is a definite dysfunction in how it's regulated. Safety-wise, absolutely should be a priority. So regulatory agencies should monitor safety and probably could do more there, maybe even in the labeling or the handling of them, for example. I mean, there was cases where people got yeast in the bloodstream because, I think at the time that the medical staff were handling the probiotic yeast and not washing their hands, and then touching an intravenous line, and so the yeast's got. I mean, there's cases where a probiotic per se has been contaminated with mold, or has caused an infection. So probably they could do a bit more with that. But in general, there's a disconnect that I would like to see corrected, and I wish the FDA would use the probiotic definition, but they don't.
I wonder if, because from what I understood from Emma, is that probiotics generally have, at least oral probiotics, generally have a high safety profile or record or whatever. And I wonder, like, governments don't really get involved in things until there's kind of a big issue, right? So I kind of wonder if, like, because generally probiotics aren't really creating a huge, you mentioned a couple examples, but it didn't sound like they're more on the oral probiotic side. Maybe governments are like, there's nothing really for us to take seriously or focus on right now, because there isn't really an issue with them.
Yeah, I mean, if you look at, there's billions of doses of probiotics taken worldwide every day. I mean, and people are not falling sick and dying from them. The irony for me is you switch on CNN or television and you see this person who has a disease, and then the next part it says we gave them this drug, and then for about 20 seconds they tell you all the things that can go wrong with this drug, including it can kill you. And then at the end of it, call your doctor and take this drug. I mean, how the heck is that? And that's because there is a benefit to this drug, but there's also massive side effects potentially. And so when you think about probiotics and the numbers needed to treat or to provide a benefit, then it's not as dramatic, because we're not, as I said, bringing Crohn's into the remission, but it's also not with the safety issues. And then the case in America where Bifidobacterium infantis strain was associated with the death of a premature baby. And so suddenly all the hospitals across the states are not allowing probiotics in the building just because there's one case. Now,
just a harsh reaction.
It's a complete overreaction. And a lot of it is based on lawyers and fear of being sued.
Mm-hmm.
And in fact, I would go the opposite way. I'd say if you didn't give my child, premature baby, a probiotic, and the child died from neck, I'm going to sue
Mm-hmm.
you for not giving probiotics.
Yeah.
I mean, it's in the States, it's really ridiculous. But yeah, it is what it is. It's as I said, without an act of Congress, it's not going to change much.
Yeah.
And in fact,
And then
we have organizations that try to have meetings with FDA, and they'll only do it in closed doors. They won't have an open, like.
why would that be?
I don't know. I mean, it's like protecting their turf or something. They're not open to just radically changing the way they do things. I mean, I'm sure colleagues of mine are better informed, and they might criticize what I'm saying. I'm just giving you my impression and opinion.
Sure.
And
yeah. I know it's true. When you watch CNN, all you see are pharmaceutical companies and their advertisements. And you're right. It's exactly the way you just described it. They talk about this drug, how it's beneficial, and then they spend so much time talking about the side effects with people laughing and smiling, and then, go talk to your doctor to how to get this. Yeah, it's exactly that, right? And then you think about how there's so much promotional use of like antibiotics even. And, we've talked a little bit about how much damage that could do. Why is an antibiotic so detrimental to the gut and the microbiome? And I also wonder, like, wouldn't the proper way of using antibiotics, I've never really got an answer to this question beyond, I imagine, just costs a lot of money, but like, why aren't we using targeted antibiotics? Like, why are we always using general strain antibiotics? Like, what's really holding us back from that? Because I imagine that would minimize the impact to our gut if we did it that way. But curious about antibiotics, how it kind of affects the gut, and then maybe your thoughts on sort of targeted antibiotics.
I mean, antibiotics have saved probably billions of lives, so I mean they're miraculous. But if you look at bacterial vaginosis, for example, the treatment is clindamycin or metronidazole. Neither of them were chosen to get rid of bacterial vaginosis. Neither of them target the organisms that cause it. Neither of them target the odor that comes from the organisms that cause it. And yet, these are the only two options, pretty much. So we've completely failed. Why would companies not then try and develop a drug? Well, first of all, I don't think technologically we have found an antibiotic that only kills E. coli. You're now starting to see people talk about phage therapy, where you have a phage that only kills E. coli. In that case, that may come to something and it may start to be a treatment. The only thing about phage is that organisms can become resistant to them the same as they can to antibiotics. But that would certainly be a target for the organism that you want to get rid of. Therefore, when you give a broad spectrum antibiotic, it's killing everything. It's carpet bombing, and it's killing the beneficial microbes as well, which, okay, if you want to get rid of the pathogen, like if you have a urinary tract infection, you have an E. coli, you want to get rid of that pathogen. But then why not at the same time give a probiotic, or afterwards for another month give a probiotic that tries to help the person's beneficial microbes that you've just wiped out, tries to bring them back. And so we actually did some work on this, where if you give the antibiotic and or antifungal and a probiotic, the lactobacilli that we gave allowed the person's own lactobacilli to come back, which is really what you want, because you don't want to replace permanently with the probiotic. Well, I mean, because it's a probiotic and it isn't going to colonize. But you want it to create an environment that their own beneficial microbes proliferate and come back. And that's what we found. So, I mean, I think that doctors are starting to do this a bit more. They're saying, we're going to give you this antibiotic. We recommend you take a probiotic. But then they don't say which one. What they need to do is go one step further and say, okay, let's look at your particular situation here. Here's the type of probiotic. It's been proven in human studies to do this and help you recover, and therefore we're going to suggest that you take that. So it's that is happening, but it's not happening to the extent that I think it could and should. And part of that is that our medical schools don't really teach people about the microbiome or beneficial microbes or probiotics. And so doctors only hear from it if they're reading the literature, if they go to a conference and they happen to be in the room where people talk about it. And pharmacists, for the most part, they don't understand it. And if I was to walk into a pharmacy with you today, we could walk right along the store. We'd find a lot of probiotics, and I would tell you that most of them haven't been proven to do anything. So why are they on the shelf? Well, they're on the shelf because there's money to be made. What I would much rather see is the companies making money. Okay, put $1 million in and do some clinical studies with your product that you're telling people confer a health benefit, and then it'll stay on the shelf. Not only that, people will come and gravitate to it because they know it provides a benefit. So there's different stages. There's the medics, there's the pharmacists, there's the companies that need to step up to the plate, and therefore the consumer then knows what they're getting, why they're getting it. The antibiotic has caused this issue, or it will cause this issue, but I'm going to counter it with this particular probiotic.
So you think there is a place for probiotics to complement antibiotics then?
Absolutely. Yeah, no question about it. Yeah. And sometimes we actually showed that you can improve the cure rate too in certain situations.
Yeah, because that was really interesting. What you said when you said that there was some finding that folks had some findings of, while the probiotic strain doesn't actually stay, it helps create the environment for the existing microbe to flourish and develop itself. So I guess antibiotics, will they completely wipe out your beneficial microbes, or is it more like if it's repeated antibiotics or a number of these kind of carpet bombs? Do we have a sense of how damaging these antibiotics actually are in removing the actual microbes that were there?
Yeah, it's pretty hard to completely wipe out all the microbes. So you're certainly going to disrupt it significantly. Disrupt it. Depends on the antibiotic. Depends on whether it's the gut, vagina, where you're applying the antibiotic. We've done some studies looking at the gut and the impact that heavy dose antibiotics will have, and the recovery of the person's own microbiome with or without probiotics. And there's definitely a benefit depending on the probiotic. In our case, there was this particular one that definitely improved the outcome. But yeah, it's none of this, which will frustrate people that maybe watch the video, none of it is just a simple, here's the answer.
Oh, it never is.
Oh, this is easy. Oh, yeah, just take this probiotic, you'll be fine. No, it's pretty complicated. But
I like what it's good to hear that you said that some doctors are starting to recognize that probiotics could complement antibiotics. Whether folks agree or disagree with this profession. My wife was actually a naturopathic doctor for about 10 years, and I know a lot of her training and a lot of her colleagues, they take that approach with antibiotics. When they know that their patients have taken antibiotics for years, they'll often have them take probiotics. I think the piece that may also be missing even in that profession is what you just said, is they might tell their patient to take a probiotic, but it's not really telling them exactly which one. And so I think there probably is some work to do. One of the
things I should also say, if you also take fermented food, that to me is a benefit, because fermented foods like kimchi, kombucha, and these things, they have a lot of beneficial microbes in them. They're not probiotics, but they have beneficial microbes in them. So I encourage people to take fermented foods to help replenish those organisms. So it's not just a case of taking a probiotic pill. You could be eating fermented foods at the same time.
That was my question about fermented foods. So just because it doesn't have those kind of four elements of what we define as a probiotic, there could be beneficial microbes in fermented foods. Okay, so that's interesting. What if could a company who's making fermented foods, could they add a probiotic to that fermented food and then say maybe that these fermented foods have probiotics? Is there a way to display it that way, or?
Yeah. So, for example, if you make a kimchi and you add 10 to the power 9 lactobacillus GG, you could potentially argue that's a probiotic. I would still say, though, I prefer you do a clinical trial with that end product.
Mm-hmm.
But yeah, see what happens though commercially. The company that the kimchi company I just talked about, they'll say, well, why would I go to all this trouble? Because on the shelf next to my kimchi is a kimchi that's been pasteurized, and it still calls itself kimchi, or it's calling the strains or the product probiotic anyway. So there's no incentive for me to say that my fermented food is a probiotic. So either you do the clinical studies to prove it's a probiotic, or you, as you say, you add a strain that you know at that concentration at the end of shelf life will give you a particular benefit.
Yeah.
But until that happens, we're not incentivizing raising the level. And what that would then take is correcting the people that continually get these things wrong, and correcting companies that say they're selling a fermented food probiotic. And I know companies that won't like that. They will say, no, we've got probiotics. We've got probiotics inside our product. Well, you shouldn't be allowed to say that. And unfortunately, you can go on the internet and there's dietitians and influencers and people, and they talk, oh, we have probiotics inside us and we have probiotics in all our fermented foods. And no, you don't. You don't. And you shouldn't be saying that. But
yeah, you might have beneficial microbes, which could be beneficial to people, but you don't have probiotics. Yeah.
No. And they wonder, why do you separate it? But it's separated because you have to define exactly what it is you have. And that's why there's now a definition for what is a fermented food and what is a prebiotic. You have to define it. It's otherwise people will just confuse and misuse the terminologies, right?
Right. And especially if you're trying to have, like, you want to use probiotics in a really clinically purposeful way, you need to have legitimacy around the term, right?
Yep. Yeah, you do.
Yep. Okay, that's interesting. Probiotics themselves, in terms of administering them, in some cases, I've read a study where with C. difficile, sometimes probiotics should be co-administered with things. Like, do you know if, like, probiotics on their own, if they're sufficient, or do they have compounds in them that ensure that the benefits are working? Any thoughts on just like co-administering probiotics with things?
Well, probiotics to prevent C. diff, I think there has been some evidence, but probiotics to treat C. diff, I think is much weaker. So once you've got C. diff, I think you need to have the antibiotics. You can have the probiotics at the same time. I think it would augment the effect depending on the probiotic. But in the case of treating, I think it's probably asking too much to get rid of a C. diff that is pathogenic and causing inflammation, etc. I don't think you're going to get a probiotic that will dislodge it and cure that disease.
Okay. And does it, so in the case of prevention though, do probiotics need to be coupled with anything in order for them to be effective?
Not that I've seen. I guess it depends. See, we don't know when we're getting C. diff, right? And how do you get, most people get C. diff if they take antibiotics. So if they take antibiotics, then you should have a probiotic at the same time.
Sure.
So that would be the co-treatment. If you want, once you have the C. diff, you're needing antibiotics anyway. So I can't think of a disease where you're taking a drug, and if you take the probiotic, it's going to do something else. And I think we need more studies like this. So if you look at a lot of the drugs in the market, they've never been tested for what they do to the microbiome. We've no idea, right? Now,
just regular drugs.
Any drug, you name it.
Yeah.
Any drug. Yeah. So why don't the companies do that? Why don't they look at what it does to the microbiome? And if it has a negative impact on the microbiome, could we and should we be supplementing with a probiotic that would counter that negative effect? So that requires careful thinking about what the microbiome disruption is, if any. And I'm sure there are many cases where drugs do disrupt the microbiome. You just need to look at the side effects. As soon as you start talking about pain and and bloating and diarrhea, then you've had an effect on the microbiota. So should these companies then recommend that you take a probiotic with it? Wow.
What's your personal approach, Gregor? Like when you if you've had to take an antibiotic, like how do you approach the situation with a probiotic? Like what do you tend to look for? Or, I don't know.
Well, I take probiotics every day anyway, but
okay.
I mean, I don't want to get into recommending specific products, but
no,
I would take probiotics that would be shown to help prevent C. diff if I'm taking an antibiotic, and have strains in it that I feel are important to replenish some of the organisms that would have been killed by the antibiotic. And I would take them during the antibiotic, and that's usually 2 hours apart. And then I would take them at least for a month afterwards.
Yeah. Okay. I want to get into the daily use of probiotics, but before that, I think there's some buzz around certain beneficial microbes, like you hear about Akkermansia and some of these other ones. Are there any of these kind of microbes that really excite you, and why?
I don't know if it excite me. I think Akkermansia is an interesting story. I think we'll have to see who it helps, when it helps, and how you use it. I think the organism itself requires acetate. I think is a prebiotic that helps it to grow. And so, should people be taking an apple cider vinegar capsules at the same time? I mean, there's a lot of interest in these. I'd say caution. It's not caution in a bad way, but let's really find out more about it and when to use it, etc. And that will be based on the clinical evidence that comes out. So I don't really see things necessarily exciting. I think probably combinations of strains are important because, as I said earlier, they complement each other. And if you have, say, I'm trying to think of the best example, but maybe you want to have some bifidobacterium and some lactobacillus because the strains that you're using have been shown to have certain properties that is helpful for the person taking them, and therefore you want more than one strain. But rather than just picking any old strain and throwing it in, we should look at the properties of that strain and then combine them. And it could be that in some people, the lactobacillus X helps them. In another person, it's lactobacillus Y. And having them both in the product increases your chances of having a benefit on the population.
Right.
But so that comes down to strategy. And I think not enough companies are doing that strategically, that they're doing it based on the species and not the actual strain properties. And I think we need to look at the strain properties.
Yeah. Let's continue focusing a little bit on sort of using probiotics. You talked about like yourself using a probiotic daily. What are the benefits of a daily probiotic? Like, it's because again, if the thought is that these things don't stick around, but then you said maybe maybe it helps create the conditions for your existing microbes to maybe replenish. We don't know exactly how it works. It seems like.
Yeah.
So why take a daily probiotic?
Well, let's go back to people that are healthy, right? So what do we mean by healthy? By the definition of the World Health Organization, nobody on the planet is healthy. Okay. Right? It's such a completely useless definition of health. You would have to be, you'd have to be a robot. But so therefore, we're not all healthy. We feel healthy. But what are the markers of issues? So in a woman who gets recurrent urinary tract infections, who's totally healthy, she wants to have fewer of these per year. So in that case, she may take a probiotic that has been shown to help prevent recurrence of infections. I might take one because my cholesterol is too high and therefore I'd like to bring it down. I'm still healthy, but I have cholesterol that's high. And this then gets back to, not only the definition of health, but unfortunately, when we do clinical studies, the persons that are recruited have to be healthy in a healthy study.
Yeah.
But if nobody's really healthy, how do you pick that? And there's people who are obese who can't get in clinical trials because they're obese. But if they're obese but healthy, they should be allowed to be in clinical trials, because that's a whole population of people that are not, we don't know the benefit of drugs and probiotics and other things because they're not included in clinical trials unless they are sick. So I think we have to wonder why it is we're taking them. So, for example, in the wintertime, let's spring. So we want to prevent or reduce the effect of allergies. Well, there's some probiotics that maybe you want to start taking in February, March, April before the allergens come in. And we did a small study in this, but other people have done studies that there will be some people that will benefit from that. That's a healthy person. You're trying to reduce the effect of that allergen on you. And so there's other cases where transit time in stool. So some people get mild constipation twice a week. Well, maybe if they took a probiotic that increases the transit time of food passing through, that would make them, they're still healthy, but it would make their life a bit easier and their stool softer, etc. So I think you could argue that depending on the person's situation, they may well benefit from a probiotic every week. Some, and I know people will argue, well, I'll just take fermented foods every week. You know what? If that does it, then that's great. But you would have to, again, look at the markers. So if you're taking a fermented foods because you like the taste, you don't want to be buying these commercial probiotic products, then do the test. So take your cholesterol level, and after 3 months, take it again. And if you found that it's gone down, then hey, maybe that fermented food was what all you needed.
Yeah.
So I think we should think of what are the things that I want to happen by taking these.
One of the things I understood about what we're seeing in sort of maybe a healthier gut or healthier microbiome is diversity across the microbiome with a lot of different species. I would imagine perhaps that a really effective a very effective probiotic probably has diversity of strains, perhaps. Again, it depends on the purpose maybe that you're trying to achieve.
Yep.
So, but in terms of a daily probiotic for general health purposes, do you think a more diverse probiotic would be helpful?
So, in order to answer that question, you need to have the clinical data to prove it. So, in theory, that makes sense. Yeah, diverse strains, but do the study. If you take this product and after 3 months, you've shown that there's more diversity in your microbiota, that all your clinical parameters, you're not feeling sick or you don't have adverse effects, etc., etc. So in theory, yes, it does make sense, but prove it. Why would I recommend you take a 50 strain probiotic unless there was evidence that, one, the strains themselves were known to have a benefit, and therefore you can be sure that there's something beneficial happening, and two, there's a study showing that it helps the diversity. And the gut microbiome is a tough thing because you'll see it in the literature. There's people write and they'll say low diversity and therefore it's bad. Well, if you did a printout, and I did this with a student about 10 years ago, maybe longer, we showed our profiles of the microbiota in terms of abundance of strains. And I said to the class, which one's healthy? I don't know. So what do we mean by an unhealthy microbiome, and how would you define it? If you increase the diversity, you would think that would be a good end goal, but how do you measure it? So
and if it's like think about David Good's research on the Yanomami peoples. they see diversity of the microbiome there.
Right.
But just because you have that exist with the Yanomami peoples doesn't mean that you can plug and play that with, you know, somebody here on a Western diet, right? And expect to get the same outcome.
Yeah. Um, I mean, in general, we think that high diversity in the gut microbiome is a good thing.
Mm-hmm.
How do you get to that is the big question. And at what age groups are we talking about?
Mm-hmm.
And so you see this thing about, you know, kids should eat dirt and things like that. What I like about the indigenous people in the Amazon and in the Hadza in Africa
Mm-hmm.
is that they eat seasonally,
Mm.
and we don't. In general, in Western society, we don't eat seasonally because we can get everything we want all year.
Right. Right.
So I think there is something to be said for seasonality eating, and I think that does help diversity, and that's probably a good thing for us. But yeah, it's, you know, I wish I could just take a paper and say, here's the proof.
Yeah. Yeah. When I think about fermented foods again, okay, no probiotics, but they could be beneficial microbes.
Mm-hmm.
Could these strains better? This is not going to sound. This might not sound proper, but could these strains better be Could they be better adopted compared to a probiotic, for instance, in the right adequate dose? Because they're kind of, I don't know, maybe there's sort of a little
Up
bit more
and running.
naturalness to it because it's coming from a food. It's not a dried state compared to a probiotic capsule or something. Like, is there better absorption, I guess, or adoption from the beneficial microbes within a fermented food?
Yeah, so I can't give a yes/no answer, but
Yeah.
I could talk about first of all, do the strains make it through the stomach and acid and the bile? Assuming they do, which you'd have to prove it, they're then getting to the small intestine and the colon, and you should be able to look at the markers, particularly short chain fatty acid markers that say, you know what, taking this fermented food, there's a response in the body that is positive, and we're seeing these short chain fatty acids that we know are beneficial.
Mm-hmm.
You would then have to then compare it. Actually, the best way to compare it would be to take the same fermented food and dry all the organisms and give them as a capsule.
Yes, yeah.
And I suspect the food matrix would be better because, as you said, they're up and living, and they're in that conglomeration, that consortium that is natural for them. And so therefore I think it would give you more markers of health benefits, but I don't know the answer for sure.
Yeah, we'd have to prove it. Yeah,
and it depends if the strains have the specific property that you want. So an example, we did some work on bifidobacterium, and there's some strains that will essentially neutralize P. cresol, which is a toxin, especially in chronic kidney disease. Well, if you have high levels of P. cresol and you take a fermented food, maybe it does absolutely nothing to those levels. In that case, you really want to have this particular bifidobacterium strain or strains, because that's an issue for you, right? So in other words, you take the fermented food, but you may want to also take a dried strain that we know has an effect that you require for your health. So
yeah. Hospitals, we know that they're really sterile environments. Do you think there is? I know you talked about your work with your first sort of Dr. Bruce there on UTIs. I imagine that would have been in a bit of a hospital setting, perhaps. But the role of probiotics in sort of these acute care settings, is there one? Do you think?
100% yes.
Okay.
So in the States, it's impossible. In Europe, they've actually
Why is it impossible in the States?
Because they don't allow probiotics into the system, and they're terrified of live organisms. So now, maybe there's some hospitals that will give people fermented food. I don't know. In Canada, we should absolutely have it. So it took a long time to persuade neonatal intensive care units to bring it in for probiotics to prevent NEC and sepsis. That has happened, and it's made an impact. even in the most at-risk patients you could imagine. I probably 10, 15 years ago, I said to our hospital, you should have 50 fermenters in the basement, and every patient that stays overnight in this hospital should be getting fermented foods with probiotic strains in them, or certainly properties that, for example, if you have, as I said, chronic kidney disease, cardiovascular disease, I would want a food that had these strains that had an effect on P. cresol. If I had diabetes, there might be a different fermented food for a different reason to affect the glycemic index. If you have a very bad inflammation, then there should be some anti-inflammatory properties perhaps. So you can have different types of foods for different reasons in a hospital. And in Europe, I actually looked up a paper last night. So this was a study that was done with probiotics and prebiotics in Europe. It says there was less fever, less coughing. It was actually, I think it was a COVID paper too. It inhibited some colicky reactions. The patients were out of hospital sooner. They had fewer antibiotics. I mean, why have we in Canada not gone to Europe and said, what is it you're doing here? And why should we not be introducing these things into our system? Because as you said, and what really struck me in the neonatal intensive care unit, it's a box. And the only thing that goes in that box are the nurses and the doctors and the visitors. So we are not introducing any beneficial microbes into that environment. We're giving all these kids antibiotics. So there's multi-drug resistant organisms. How do we get beneficial microbes into that environment? The only way is through a probiotic. Now, you could say the same in a hospital ward or a place where patients are. Why on earth are we not doing that? We're very happy to give the toxic drugs, and yet we won't give beneficial microbes. So I think we have to get people to do that. You know, go back to the funding again. Like, why are we not funding studies in this in hospitals across Canada? And looking at, imagine if you had the patients out quicker, if you had less infections, if you had fewer side effects of the drugs. The benefit is there for everybody. We're not doing it, not funding it.
Well, and I imagine so many of these babies in the NICU are probably not getting delivered vaginally, right? Like they're probably, they're probably C-section, so they're missing the whole mother's, vaginal flora and all that. So, you make a good case on that. That brings me to my next question is for parents and their children. Like we know that the microbiome pretty much gets set, I think, by the age of 5 or something like that. Like within the first, several years of life, generally. Like, I mean, you talked about how you could maybe influence that with probiotics, but my understanding is generally it gets established pretty early on in life. What should parents consider in terms of helping develop their children's microbiome? What are the factors do you think they should consider?
It's a very hard question to answer because every child is different. Some are born cesarean, some are born vaginally, some get antibiotics, some don't. Vaccines have an effect. I mean, should you eat dirt? I don't think so. But should we have kids playing in around nature? Absolutely. And if we know there's no pesticides or toxic things that are around, then I think it's good to have kids do that. You need to have organisms that synthesize vitamins. Do we check to see if the kids have that? And if they don't have it, should we be giving a probiotic that stimulates vitamin production? If they're breastfed versus formula fed, that's going to have an effect. So if mom can't breastfeed, then you're giving a formula. Should we be giving a formula with a probiotic? Unfortunately, there's only formulas with one strain of probiotic in it. I'd kind of like to have seen maybe one with multiple strains, but should we then be supplementing the kid with a probiotic product, say at age 2, 3? I mean, these are lots of questions. We are just, in my opinion, starting to look at the timing of foods. So when should I give my kid eggs and peanuts and fruit and things like that? So in an ideal world, you would have a study where you really follow kids, look at when you're introducing different foods, what their microbiome is like, what their metabolites and vitamins are like, what their lifestyle is like. Are they out playing in the country? Are they just in a daycare where there's 50 kids and all they get is colds and flus and get sick all the time? Even that we should be studying, because people say that's a good thing in the long run. It helps your immune system. Well, like 10 colds a year, is that a good thing, or is that too much? Is it? So I would I go back to the possibility of human studies. This is another example. It would take a few $1 million, but that's what we should look at. And you're not going to come out with definitive things because every kid is different. But I think in general, we'll start to understand. We're starting to see what microbes are appearing at different stages in vaginal born versus cesarean, breastfed and not breastfed. That's great. Now we need to look at the vitamins and the minerals and other, like the metabolome of those kids.
Right.
Then we look at the lifestyle and the food that the kids are eating. So it's a great longitudinal study to try to start answering your questions. It will probably raise more questions than answers, but I think it would be really valuable, because right now we don't know anything. I mean, I show when I was teaching, I showed a slide of a developing baby, and the heart develops very early, and then the brain starts to develop. What have we done for that child at any of these stages in terms of microbiome or food? The answer is pretty much nothing. I mean, we give some vitamins maybe, and as I said,
You're talking about from the mother's perspective, like
no, from the baby. Well, first of all, in the mother in the womb, in the mother's perspective, what have we done? Nothing. And the microbiome and the metabolome will affect that baby. So what have we done? Nothing. We don't understand any of it. And you've seen the studies in particularly Finland where they'll introduce a probiotic to try and prevent that child from having an allergic ectopic response when they're born. So that's one of the examples of trying to affect the pregnancy in a positive way. But in terms of food and microbes, we haven't looked at that and how it affects the development of that child. And to me, that's, again, a big gap in our understanding. Of course, it's very difficult to intervene because ethical issues, and we don't want a thalidomide. But we could at least start with monitoring the nutrient intake, the microbiome composition, and then as the baby's born, the environment they're in. And there was a beautiful study on sandboxes from Scandinavia where this guy showed that, some sandbox can't have ones with pathogens, but some of these kids may eventually have fewer allergies and better health if they're allowed to go out and play with microbes in the environment. So complicated answer, but it's a complicated issue that, again, is not being funded, which to me is if we really are a Canadian Institute for Health, why the heck are we not studying those types of parameters in the development of every human being that comes into the world?
Right. We should be open to all of it for sure.
And if someone's living in a city, you go to China, everybody's living in a skyscraper. That's going to be a different environment too. And maybe there are things in the food or in the environment that we can help supplement those babies so that they do get exposed to different organisms and have better diversity.
Yeah. One of the things I wonder is post-COVID, whether kids are developing a fear of germs because of all the, use of hand sanitizers and all the precautionaries that we're giving to kids, which, was obviously, appropriate during the COVID period, but what are the long-term effects of the psychology around how people think around? I just think of my own daughter. She's 6.5, and she struggles sometimes with, like, she feels like she always has to wash her hands, and she always feels like she has to anything that could be dirty, like, that's in her head a little bit. And we try to right size that for her, but it's tough. So
it is. Yeah, I hadn't thought of the COVID impact, but for sure. It's I think it impacts the parents too, because you're constantly worried about a virus that you can't see. And it's, of course, COVID's still around. But the only way is to try and re-educate kids that it's okay to be in, to pet dogs and to have exposure to dirt and different foods and stuff. But it's difficult because we've created an environment, in my opinion. If you look at how we grow chickens and pigs, so we grow chickens basically so that they have salmonella, right? So then when the chicken meat comes back, we have to be careful how we handle it. We have all kinds of drugs in Canada. It's over 100 drugs in our water supply, in our wastewater treatment plants. We have fields where we spray pesticides. So we're pretty good at creating situations where you want to kill the salmonella, or you want to avoid exposure to these chemicals, and that's probably not how. Well, certainly not how the indigenous people in the Amazon and in Africa are living. And so I think a lot of it is self-created by humankind and how we have created society. So how do you get a kid to kind of go back to thinking it is okay to be around germs, but don't touch the chicken that's on the counter, and don't go into that field after it's been sprayed by pesticides, and don't go into that standing water because there might be giardia in it. And so it's not a simple let them go and enjoy the environment. But re-education. But has your kid ever learned about microbes in school?
Not that I know of. No, not yet. I mean, she's starting grade 1 now, but I mean, they obviously talk about how to prevent the spread of disease and all that sort of thing. Like, that's very prevalent, but nothing about like that there are also good germs and so I mean, we're trying to tell her, but it's not. I don't know how much of it is landing. You talked about how we have this environment where we create this kind of disease environment, and then we have solutions to combat that. I mean, that's also, there's a business proposition to that too, right? Of course. So I'm recognizing time, and I know I'm pushing our time here, but there's one thing I want to ask before I get into some really quick sort of rapid fire questions that I saw, just people top kind of search questions that I thought would be interesting to fly by you. But you folks did some research on mercury and probiotics, right? Do you want to just quickly speak about, like, it sounds like probiotics had an influence on potentially the absorption of mercury, if I'm understanding that correctly. Do you want to just quickly speak on that, Gregor?
Yeah, so we were in Africa with a project where we taught local mothers how to make probiotic yogurt. And if you try and picture the city, so it's Mwanza, it's on Lake Victoria with 40 million people living on the lake. And one of the things that happened was that women, we're talking about very poor people, and women, it turns out, got HIV because they'd go down to the harbor and they would basically have sex for fish. So there was this sort of association with fish and women, and I thought to myself, well, never mind the HIV component, what are these fish like, and could there be mercury in the fish or arsenic or other things? And it turns out that a lot of people were panning for gold, and they were using mercury to pan for the gold. And so the mercury was going into Lake Victoria. And when we looked at the fish, the small silverfish, were really caked in mercury, basically. So these women were going down and they're getting the fish, bringing back, cooking up for their families, and they had high levels of mercury and arsenic. And we did a study in the lab and it found out that these lactobacilli strains could bind to a lot of these heavy metals. So we then thought, well, if they bind to them, I wonder if we could do a study where you give the probiotic yogurt, which has the lactobacilli, the GR1 strain that binds to mercury, and see if you could reduce the absorption of the heavy metals. And it turned out in the kids and the mothers, there was evidence of that. So then you think, well, what about in Canada? I mean, it's, obviously the Sarnia native people have had big issues with mercury. We haven't brought those concepts back to Canada. It very much frustrates me that it's good to fund work in Africa, but then it's like, whoa, we're helping the Africans. Well, wait a minute, can't we then help the Canadians? Couldn't we do a study where we look to see if giving strains of, say, lactobacilli that bind to mercury and arsenic and lead, etc., could somehow help our people? Because once you've got the heavy metal in you, it's hard to get it out. So you're trying to reduce the uptake of it, and that's where that's how it came about. And it then also led to the interest in pesticides and then the honeybee work, because we were now talking about environmental pollution, and therefore that's what led to Brendan doing nice work on the honeybees.
Yeah, that's very interesting. And I imagine that, I know talking to David Good about right now, the indigenous peoples in the Amazon, they're facing obviously a lot of gold mining and pollution of their rivers and water systems with mercury. That would be interesting to apply it to other areas like that too. Very cool. Couple of really quick rapid fire questions, and then we can kind of wrap up if you're okay with that. So these are some of the top search questions that I found that people had. So are expired probiotics still good?
Well, when we say expired, we mean that the end of shelf life, so you shouldn't really take them. Having said that, we did a study, we actually published a study showing that there were some organisms alive even quite a while after expiry date. So therefore it depends on what should really happen is they should do a study that says, say 10 to the 9 will give you an effect, but 10 to the 7 will not give you an effect. Therefore, at the end of shelf life, if you're now down to 10 to the 7, you shouldn't take that. But because companies haven't done that, what do they mean by end of shelf life? How many are dead and how many are still alive? So you don't know the answer. Therefore, I would say don't take them. Yeah. But there's probably organisms still alive in it. Depends on how they've been handled. I mean, if they've been left out in the sun, or they've been left in the air, or the organisms have had moisture, and then they can grow and multiply in the capsule and therefore die. But I would say no, don't take them.
Yeah, yeah, and that goes back to like having the adequate amount too, right?
Yeah.
I recognize doing rapid fire with an academic is not always going to work really well. So
I was supposed to do that quicker. Sorry about that. I should just have said no.
No, I like the nuanced answer. It's good. Can probiotics cure bad breath? Is also a top search question.
I don't know they can cure it, but there's a couple of probiotics that can help alleviate bad breath, but I haven't seen anything that's cured bad breath.
Okay. Probiotics for acne. People are interested in that.
There's some nice work being done on acne, psoriasis, these types of things. Curing it, I'm not so sure, but having an effect on it, I think there may be some data on that. So I'd be optimistic, but how would you apply it? So there's the theory that you have to apply it orally because there's an immunological reaction to some of the, psoriasis. In terms of acne, do you apply it locally? In which case, what strains are approved to be applied locally? I'm not sure there are any that are being approved as a cream. So you'd have to do some research to find out which ones. I mean, we did studies on wounds, and showed that you could apply a probiotic directly to a wound and help with the wound healing. But once it's okay to do an experiment like that, but to actually have that approved by regulatory Health Canada, because you don't want to get an infection through an open wound. So I would say there's probably something going on out there, but it's not a magic bullet yet.
Yeah. Okay. Are probiotics good for bacterial vaginosis?
Yeah. Yes. So there's the strains that we worked on. I've no conflict of interest with that now. And there's a new product out on the market from Seed, and I'll declare that I consult for Seed, but it looks promising. It's a Lactobacillus crispatus vaginal product, and the studies so far suggest that the strains are really good. But again, you have to rely on the clinical data being published and reviewing it, and that hasn't been done yet. But it looks promising, and I think in the probiotic chart there may be a couple of other products that show some promise. So yes.
Can probiotics cause SIBO, small intestine bacterial overgrowth?
Not that I'm aware of. I think a long time ago there was a study suggesting you could treat SIBO with a probiotic. I think it was NCFM was the probiotic that they used, but I haven't seen any evidence that you would get SIBO because of taking a probiotic.
Yeah, great. I usually have two questions I ask every guest. They're kind of fun. Did you want to get through them?
Sure.
Okay, awesome. So our 5 for dinner question, dead or alive, who are 5 people you would want to have dinner with?
So it's good that you gave me a couple of days to think about this, because I asked some students, we had a lab party the other night, and for the life of me, none of them could come up with even one name. So I was pretty disgusted with them. So my 5, Denzel Washington.
Denzel.
Wow. Yeah, I think he's an amazing actor. I think he's a really interesting person to talk to.
Favorite Denzel movie?
Man on Fire.
Man on Fire is a great one. I like that. Yeah, I've watched that many times. Yeah.
Yeah, Equalizer's good too, but Man on Fire, the music was fantastic too. Yeah. Sir Alex Ferguson is the manager of Manchester United, and he was manager of Aberdeen when they were the best team in Europe, which is unbelievable. I think he would be an amazing person. He actually wrote to him, so it was funny. I was in Bermuda. I was watching a game of soccer in the golf club, and the guy next to me, we got chatting. We're watching this game, and I said, we got talking about Sir Alex Ferguson. I said, I've always wanted to write to Sir Alex and thank him for what he did for Aberdeen Football Club. The guy turned around and he goes, I'm his neighbour. He says, why don't you write the letter and I'll deliver it to him? I said, you're joking. No. So he says, so I grabbed a piece of paper from the bar and I started writing. My handwriting is terrible, right? Yeah, so I wrote this, it's like three-page letter or whatever. Anyway, but 3 months later, I got a reply from Sir Alex saying, thanks for the letter. And he says, because he was brought up in a pretty rough part of Glasgow, and we had some experiences in Glasgow that weren't the best. And so he could relate to that. He could relate to my dad, and it was really nice letter. Anyway, so that's number 2.
That's pretty close, though, to, like you had a little bit of an exchange with him.
Yeah, super. Yeah. Number 3, Taylor Swift.
Okay, wow.
It's a couple of things I'd like. First of all, how she writes songs. Some of her songs are fantastic. And how does she go about it? Does the lyrics come first? Does the tune come first? How does she cope with essentially not being a normal person anymore? I mean, private jets and stuff. So I think it would be very, very interesting. Jesus, I would love to know more about who he was as a person, and there's so many questions you could ask. And then the last one, it was actually I thought of Margaret Thatcher, because she's hated by so many people, and yet she did some amazing things. And she also had the Falklands War, and I was almost involved in that because I just finished my PhD and I was going through South Africa on the way home to Britain. And the Belgrado was a large warship that was blown up by the Brits in retaliation for the Sheffield being bombed, which is a warship. So after this large ship was destroyed, the Argentinian Navy backed off into Argentinian waters, and the story was that there was 2 British nuclear-powered submarines basically sitting watching them, and were threatening to blow them up. And if that had happened, if they'd attacked, then both countries would have to have declared war. They hadn't declared war at that point, and everyone at my age could have been called up. So I was sitting in South Africa thinking, do I really want to go to Britain, or do I just want to fly straight to Canada? Because I'm going to get seasick going to the Falklands in a ship. And then, no offense, I didn't feel like dying in the Falklands Islands.
Sure. Yeah.
So I'd like to have talked to Margaret Thatcher about all that sort of stuff. But I've taken her off the list because I'm only allowed 5. So Jesus, Denzel Washington, Taylor Swift, Sir Alex Ferguson, and probably my dad.
Your dad.
He passed away, and now I would love to see him again and talk about a whole bunch of things. So that would be my 5.
Yeah, that's a great 5. I remember watching, did you see the Beckham documentary on Netflix and his relationship with Alex?
Sir Alex?
Yeah.
He threw a shoe
very
at him.
Interesting.
Yeah. Well, he was like a father figure to Beckham, but then Beckham got into fashion too much and Sir Alex kicked him out.
Yeah.
Which is, he's quite right, because the priority should be football, not fashion and tattoos.
Yeah, fair. Yeah. The Taylor Swift thing. I mean, you're probably the 3rd or 4th academic who has said Taylor Swift, so there's something there.
Well, I also thought about Chris Martin of Coldplay, because again, for the songwriting and how he goes about it would be amazing. But I wanted to make sure there's a woman in my 5, so that's why Taylor got the vote.
Yeah. Usually I can draw a connection across the 5, and it helps me understand the person behind, like you essentially, but I'm not I can't really find the interconnection amongst these 5 and what the theme is overall. So I'll have to take that away and think about it.
But that's, I've always liked different things.
Yeah.
I mean, when I go to concerts, I want to be in the mosh pit at the front row, even at my age. So, I have diverse interests, and I try and encourage them. That's why I wrote these crime novels, because I like writing and thinking about different things. So, yeah.
I feel like you're the kind of guy who would be great to have a beer with. Although I have a story when I went to England one time when I was 18. My cousin, he owned a pub, and he let me serve a few beers behind the bar. And there was an Irishman and a Scotsman. And so I served the Irishman, and I served it right to the top. And he got mad at my cousin Raj. He's like, Raj, what is this guy doing? And Raj told me, he's like, the Irish like a little bit of head on their beer. So next guy comes in, Scotsman, had no idea. Filled it with a little bit of head on the beer. Scotsman's no, what are you doing? I have to have every inch of that beer. And Raj is like, the Scots, they're a bit cheap. They want all their beer that they can. So a little bit there, but yeah.
Yeah, well, I've got Irish stories too. My brother married an Irish girl in Galway, and my best friend from New Zealand came, and they were having a Guinness, and he said to the Irish guys, "The nice thing about Guinness is you could never really get drunk with it."
No.
And everybody looked at him and said, "You off your head?" What he meant was it was such a stout that you couldn't drink enough because you'd be full up. Anyway, 5 minutes later, he had 7 pints of Guinness put in front of him, and he said, "Let's do the experiment." He never finished them.
Yeah, that's hilarious. Last question. Besides the circle of life, what do you know for sure?
That's a really very hard question, because it's for sure. I think probably the answer is for sure we don't really understand life in the universe, and I wish we did. And so with that, it's, where did we come from? Where did we go back to, if we do at all? And what is out there in space? Because the Webb telescope is sending these amazing pictures, and it's only a pinhead of what is out there. It's just maybe our brains are not big enough to understand it, but for sure there's something amazing going on that we don't understand, and I don't know when we will understand more of it. But it's kind of not the best answer to that question, but it's the only one I can come up with.
Yeah, that's a great answer. And the James Webb Telescope, what an incredible project! The number of things that they had to get right. I think they said there were like 300 and something plus possibilities of it going wrong, and for them to make sure that it all got right. And then it's like launched out there, which they've never been able to get there. Anyways, it's amazing, incredible.
It is amazing. It's beyond the scope of understanding, really.
Totally.
It's amazing.
Yeah. Gregor, this was a fantastic conversation. It was everything that I was hoping it to be. All things probiotics. I think we hit on a number of different topics. I really loved your conversation with Rich. I thought that was fantastic. So I was hoping to build off of it, and I think in some ways we did. So thank you so much for making time for me. Really appreciate having you on the show. You're welcome back at any time, of course. And just enjoyed chatting with you.
Well, I didn't really get the definitive answers that you wanted, or that people wanted. So I apologize for that. But I think in part it's because there's no real definitive answers in a lot of this. And I think all we can do is try and understand it as best as we can, and follow the literature, because it's the data that tells us, not the words. And so the more research that we can get done, the more results that tell us what works, what doesn't, and why it works, then the better that will be. And so maybe some of your listeners will end up being the people that drive this forward, which would be kind of cool. And maybe even the lay people who feel like that science is beyond them, maybe they can still contribute in different ways. So thanks for having me on, and good luck with your podcasts.
Thank you so much. Thanks everyone for joining today. Like, subscribe, do all those wonderful things, and we'll see you on the next episode. Cheers.